Welcome to P.Bright Academy. Today we shall discuss the second part of the ISRO VSSC Pharmacist Exam solved question paper with detailed explanation. The exam was conducted on December 2017. Moving on to the question, the pH of a pharmaceutical buffer system can be calculated by. Options are pH partition theory, Noyes-Whitney law, Henderson-Hasselbalch equation, Michaeli-Mendez equation. The right answer for this question is Henderson-Hasselbalch equation. So let us see what the other options are, as well as what is Henderson-Hasselbalch equation. Okay, Henderson-Hasselbalch equation is basically to determine the concentration of unionized and ionized drug. So using Henderson-Hasselbalch equation, we can determine the concentration of unionized or the ionized drug, either for a weakly acidic drug, or it can also be for a weakly basic drug, depending upon which the equation for the Henderson-Hasselbalch equation will change. So whether it is for a weak acid, the equation becomes pH is equal to pKa plus log concentration of ionized form divided by concentration of unionized form. So if it is for a weak base, the equation becomes pH is equal to pKa plus log concentration of ionized form divided by concentration of ionized form, that is log concentration of unionized form divided by concentration of the ionized form. Here pKa indicates the ionization constant, or it is also known as the dissociation constant. Okay, so another important question which we usually see from this session is which type of drug in what form will be absorbed the most. So we know that the drug is of two basic types, right? One is the ionized form and the other is the unionized form. So we have two different forms of drug. One is the ionized form and the other is the unionized form. So ionized form of drugs, or if the charge is present in the drug, then this type of drug is not absorbed into the systemic circulation, or else we can say that it is not absorbed through the gastrointestinal tract. Okay, it is not absorbed. But if it is in the form of unionized drug, this will get absorbed through the GIT tract. Okay, so once it is absorbed through the GIT tract, then it gets absorbed into the systemic circulation, or we can call it as the blood circulation. So drug, if it is present only in the unionized form, it will reach the systemic circulation. If it is in ionized form, it will not get absorbed through the GIT. So this is an important question for different competitive exam. In what form the drug gets absorbed into the systemic circulation or through the gastrointestinal tract? And the answer for that is, it is as unionized form. Next, let us see what the pH partition theory tells us about. In pH partition theory, it is talking about the absorption of drugs. Okay, so the absorption of drug, it is depending upon different factors such as gastrointestinal pH, ionization constant, or we can call it as the dissociation constant, and as well as the lipid solubility of unionized drug. These are the different factors which is influencing the absorption of drug. Okay, and that is what pH partition theory is talking about, right? And we have already discussed in which form the drug is getting absorbed, which is an important MCQ question. This is also has been asked in different competitive exams, pharmacist exams and all. Next is Noyes-Whitney law. This is basically for dissolution. Okay, so let us just check what is the definition of dissolution. We know that dissolution is a process by which solid substances enters into a solution, right? So that is what dissolution means. And here the Noyes-Whitney law, it talks that the rate of change of concentration of a dissolved material with time is directly proportional to the concentration difference between two sides of the diffusion layer. So this is what Noyes-Whitney law is talking about. It is basically related to the dissolution process. Okay, we have to more give importance to the principle, that is the dissolution. Next is Michaelis-Mentes equation. So we know that Michaelis-Mentes equation is basically related to the enzyme substrate reaction. So we know in order to catalyze a reaction sometimes we require enzymes, right? So enzyme catalyzed reaction, the rate is discussed in Michaelis-Mentes equation. Okay, so this is basically for an enzyme substrate catalyzed reaction. Next question, the Pharmacy Council of India is constituted by, we have different options: central government, state government, parliament, legislative assembly. The right answer for this question is central government. So the Pharmacy Council of India is constituted by the central government. So there is another important session which we have to discuss. Okay, that is DTAB. As you all know, DTAB stands for Drug Technical Advisory Board. This is another important area where usually questions are being asked. So what is DTAB, and the number depending upon the actual number of members present in DTAB, these are all areas of interest. So total number of members which you can find in DTAB or Drug Technical Advisory Board is 18. Okay, this 18 figure is actually divided into 3: ex officio members, nominated members, and elected members. So first we'll see who all are coming under ex officio members. So total of 8 members are coming under ex officio member. The top position is by the Director General of Health Services. He's also acting as a chairman of DTAB. This is very important. The question will be asked like, who is the chairman of Drug Technical Advisory Board? The answer for that is Director General of Health Services. Next member is Drugs Controller of India. Okay. Next is Director, Central Drugs Laboratory. Another area of interest usually being asked in various competitive exams, the place where it is located. So Central Drugs Laboratory is located in Calcutta. Okay, and Director of Central Drugs Laboratory is an ex officio member of DTAB. Next, Director, Central Research Institute. So where it is located? It is located in Kasauli. Okay, this is also an area of interest. Next, Director, Central Drug Research Institute, and this is located in Lucknow. Okay. Next, Director, Indian Veterinary Research Institute. This is located in Izat Nagar. And the other ex officio members are President, Medical Council of India, and President, Pharmacy Council of India. So total we'll have 8 ex officio members for DTAB. Okay, or we can call it as Drug Technical Advisory Board. Next, we are talking about the nominated members. So who all are the nominated members? Total of 5 members will be there as nominated. 2 persons, they are nominated by the central government from amongst the person who are in charge of drugs control in the states. Okay, so at state level we'll see who is the the person who's in charge of the drugs control of the state, and from all the states we'll select 2 persons. 2 persons will be nominated by the central government. So it is not the state government who is nominating, it is the central government who is nominating 2 persons from among the person who is in charge of drugs control in the state. Next, we have 2 government analysts. Okay, this is also nominated by the central government and not the state government. Okay, so both these nominations are coming from the central government, and 1 person will be from the pharmaceutical industry. So industry, one person will be nominated again by the central government. So you can see all the nominated persons are by the central government. Next, going to the elected members. So we'll have total of 5 elected members in DTAB or Drug Technical Advisory Board. A teacher, basically from the pharmacy background, either from pharmaceutical chemistry or pharmacognosy or pharmacy, who is a staff on the Indian University or affiliated colleges, and is elected by the executive committee of PCI, okay, Pharmacy Council of India. One person who is elected, one from the teaching field. Next, a teaching field teacher who is basically from the medicine or therapeutics field, okay, again who will be elected by the executive committee of medical research, okay. Next, a pharmacologist elected by governing body of the ICMR, that is Indian Council of Medical Research. One person who is elected by the Council of Indian Medical Association, or we can call it as IMA. Then one person who is elected by the Council of IPA, that is Indian Pharmaceutical Association. So total of 5 members are the elected members, 5 members are nominated members. They are nominated by the central government, and then there is ex officio members, total of 8. Okay, so these are the important points which you have to remember when you are studying Drug Technical Advisory Board. Let's move on to the next question. Poorly manufactured tablet may have small pinholes on the surface. This phenomena is known as. Options are picking, mottling, leaching, and cracking. The right answer for this is picking. Okay, so this is an area of interest for different competitive exams. We have to know what are the different defects of a tablet. Okay, we know that the manufacturing of tablet involves different process, and there will be different tablet defects, which we can divide into 3 basic types, okay, depending upon whether it is process related, whether it is excipient related, and whether it is machine related. So this is a basic category of tablet defects, problems which we face during the tablet manufacturing, either process related, excipient related, or maybe related to the machine being used. So let's see what are the process related defects. One is capping, second is lamination, third one is cracking. So what is the excipient related defects? It is chipping, sticking, picking, and binding. What about machine? Machines, it is double impression, okay, double impression. So these are the different tablet defect. Now we have to see one by one, because this is an area of interest for different competitive exam. Let's see what is capping. So as the name indicates, there is formation of caps, okay. So the upper and lower segment of the tablet can separate horizontally, and it gets partially or completely, it gets separated from the main body of the tablet, and it just comes out as a cap. This is called as capping. So if you observe here, you can see that this leaves as a cap, exactly like a cap. It leaves and it's getting divided like horizontally. That is the first type of defect, which is called as capping defect, okay, and this is process related. Next, what you see is the lamination. So lamination, as the word indicates, it is a formation of different layers. In particular, we call it as distinct horizontal layers. So you can find here that there is different horizontal layers being formed, which is known as lamination defect. So this is lamination defect. So let's see what is the next defect. It is known as the cracking. So what is cracking? Cracking means the upper and the lower center surface of the tablet cracks, or you can observe that there is crack on the top side or on the upper side. Similarly, on the lower side also you will find different cracks being formed, and in very rare conditions, even on the sides of this tablet you will be able to find cracks. This is what's called as cracking. So all this is coming under the process defect. Now let's see what are the excipient defect. First one is chipping. So in chipping, the tablet edges. So as you can see, the tablet edge breaks off. Okay, and this is basically happening during either when the tablet leaves a press or subsequent handling or during the coating operation. Okay, so basically chipping means the tablet edge will be breaking off and leaving from the surface of the tablet. Next is sticking. So sticking is again related to the excipients. So sticking, as it refers, the tablet material will be adhering to the side of the die wall. As you can see, this is a die here, and on the side of this die you will be able to see the tablet materials. Okay, so from the tablet materials, if it is not properly formulated, it will get sticked onto the side walls. Okay, side walls of die. So this is important. It is a side wall of the die to which the tablet material is getting sticked onto. Okay, this is called as sticking. And next is picking. So as the term indicates, picking means the die, when it goes out, the tablet material, small amount of tablet materials will get sticked onto the die and will be removed from the tablet surface by the punch face of the die. So this is the punch face of the die, and when the die is removed from that region, from the tablet, small pieces will get adhered to the surface of the die. So what happens to the tablet is that it will result in a development of pinholes. Pinholes means on the surface of tablet you will be able to see small gaps. This is called as pinholes. So in which case pinholes is developing? In the case of picking, pinholes is developing. That was a question asked in this competitive exam, right? So let's see that question once again. Poorly manufactured tablet may have small pinholes on the surface. This is because of the picking defect, right? So next is binding. So what do you mean by binding? The tablet will adhere to the die and it just moves away from that surface. So as you can see here, the tablet is now attached with the die. This is what called as binding. Okay. Next, mottling. Mottling is the unequal distribution of color on the tablet surface. So if you observe here on this picture, you can see that some areas are dark and some areas are light. So you have alternate dark and light spots on the tablet surface. This is what called as mottling. So mottling means unequal distribution of color, especially with the formation of alternate light and dark spot on the surface of the tablet. Okay. Next defect is the machine defect, which we have already seen. It is a double impression. So here the punch, it is freely movable, and we know that on the punch surface there will be sometimes monogram which get engraved on both the surface of the tablet. Okay, this might be because of free movement of the punch, and which results in a double impression. So you will be able to see monograph on both the surface of the tablet. This is another defect, mainly because of the machine being used. Okay, so thank you for watching our channel. If you like the video, please like, share and subscribe to our YouTube channel, P.Bright Academy, and also you can follow us on Facebook for more educational videos. Thank you.